with my channel Solving Questions with Gauri ma'am. Today we will solve one question from the TIFR GS exam 2016. When performing X-ray crystallography to determine the protein structure, the scattering of X-rays from the protein crystal is primarily due to options are amino acid, carbon atoms, electrons or protons. So the question is about X-ray crystallography. Let's understand what is X-ray crystallography and find the answer for the question. X-ray crystallography is the technique which is used to determine the structure of protein or nucleic acid also. Here the compound whose structure has to be determined is taken in a crystalline form. And why X-rays are used? Because X-rays are having wavelength similar to the interatomic distance in a particle. So here I have written the wavelength of X-ray. It is 0.01 to 10 nanometer. So with this wavelength, X-ray can pass from in between the two atoms, and once it hit the atom, it gets diffracted. So this diffraction takes place due to the electrons and not the nuclei. Okay. This diffracted ray scattered around it. This is recorded by the photographic plate. If they reinforce each other, they are in phase, then we find the dark band. If they cancel each other, then the band is light one. So this way, using the Bragg's law, X-ray diffraction is studied and the protein structure or nucleic acid structure can be determined using this technique. So remember that electrons are responsible for the scattering of X-rays and not the nuclei of the atom. So with this discussion, we find that option C is the correct answer for this question. Electrons are required for the scattering of X-rays from the protein crystal during the X-ray crystallography. Now think about other options. We know that amino acids are the building blocks of proteins. Using the amino acid, a long chain of polypeptide can be formed. These are used in the translation during the protein synthesis and that are linked with a peptide bond to form a polypeptide chain. There are 20 amino acids which get incorporated into a proteins. The properties of these amino acids varies according to the R chain which is present in amino acid. That gives the different properties to amino acid. Secondly, there is a option carbon atoms. So this is carbon atom is the main atom which is present in all biomolecules. If we see the four biomolecules routinely we study proteins, nucleic acid, then carbohydrate and lipid. In every biomolecule we find carbon atom as a main atom. So again, if we perform a X-ray crystallography, same thing happens here also. In the atom, due to the presence of electron, the X-ray scattering occurs. We have understood about the electrons, and the last option is about protons. So protons are subatomic particles. In the nucleus of the atom, we find proton and neutrons, and around the nucleus, electrons are revolving. So these are positively charged particles which are present inside the nucleus of atom, and negatively charged electrons revolve around the atom, around the nucleus in the atom. So this is again, do not have any concern. of the diffraction of X-rays during the X-ray crystallography. I hope you like my videos. Please don't hesitate to like, share and subscribe for my channel, and also to hit the bell icon. Thanks for watching.